Okay, everyone. Hi. Thank you for joining us today. This is another one of the SimSip scientific webinars. And you'll see today that we've got Sarah Quinney from Indiana University School of Medicine. So she's going to be doing a talk. Just before she starts, I'll just do a bit of housekeeping. Obviously, if you can raise your hand if you have a question, and I think we'll come to them at the end of the presentation, Sarah, if that's fine with you. And yeah, but any questions, you can either type them in the conversation, or you can raise your hand and we can address them. There'll be time at the end for questions. Sean, have you recorded? In terms of recording, it should be available after this presentation today. It'll be uploaded later on. If we go on to the next slide, sorry, Sarah. So Sarah is today's speaker. She is Associate Professor of Obstetric and Gynecology and Medicine, and also a Director of the IUSM Disease and Therapeutic Response Modeling Program. And she's obviously highly involved in maternal and pediatric precision therapeutics. You can see she's a PI on this center, and this Model Knowledge and Research Coordination Center. So we've asked her to present on a paper that was BCR RP, I think it was, published. And it's an important paper, not only because it covers special populations and pregnancy, obviously a population with high need for dosage recommendations, but also an important part of this paper is looking at model reproducibility. Specifically, you're looking at different versions of the SimSip simulator, and how also it's important to have that reproducibility. So I think you're going to take over now and lead us in the presentation. So thanks for agreeing to speak to us today, and looking forward to it.
Thank you, Oliver, and thank you for inviting me to give this talk today. So just move on into it. So just a brief outline of my presentation. First, we'll talk a little bit about physiological changes in pregnancy and how PBPK modeling can help inform drug dosing in pregnancy. But then the majority of the talk will be really focused on some experiences we've had in trying to reproduce PBPK models. I'll give a couple of case examples of reproducibility issues we've faced in trying to recreate models in pregnancy, and also to discuss some steps and best practices that can enhance PBPK model reproducibility. So one thing I'm often asked when I talk about being an obstetric pharmacologist is, well, pregnant women don't use a lot of medications, so what is there to learn? Well, that's one of the problems, is that people don't think pregnant women use a lot of medications when they actually quite frequently use medications during pregnancy. This data is actually a little bit dated. It's from an article published in 2011 on overall medication use in pregnancy women. And you can see that from the 1970s through 2008, the number of drugs taken anytime during pregnancy, indicated by this purple line here, has increased, or sorry, the percent of individuals taking medications has increased. and the percent taking 4 or more medications has also increased. So about 20% have took 4 or more medications in the early 70s, but now up to 50% take multiple medications or at least 4 medications during pregnancy. And there's differences based on age, education status, with those who have completed high school tending to take more medications during pregnancy, and also race and ethnicity, with non-Hispanic whites having a higher number of medications than Blacks, Hispanics, and other populations. This is not just shown by this single study. Recently, IU participated in recruitment for the New Mom to Be study, which is a study of almost 10,000 nulliparous pregnancies that took place. Recruitment was from October 2010 to September 2013 across 8 geographically and ethnically diverse centers in the United States. And in this study, most of the medication use was self-reported, although we also collected data from chart reviews. But you can see that the majority of women in the study took some sort of medication. About 70% took at least one medicine besides a prenatal vitamin, and 30% took more than 5 medications. And you can see on the right-hand side, the list of medications varies from things that are used to treat complications of pregnancy, like tocolytics and progesterones, to drugs that are used to treat common conditions that continue to have that pregnant women also have issues with, analgesics, antibiotics, antidepressants, thyroid medications. So women are still people and still get illnesses, and so they'll need to have medication treatment for that as well. But women, a lot of times we are left with extrapolating information on medication dosing in pregnancy from what we know of healthy volunteers or non-pregnant individuals. But there's a lot of dynamic changes that occur during pregnancy that can really impact the pharmacokinetics of a drug. And these changes happen across pregnancy and are not just static throughout gestation. For instance, maternal weight will increase. The fetal part of this is due to fetal growth and volume expansion. But we also see then with a change in plasma volume, and so you see a decrease in albumin concentrations, changes in renal blood flow and glomerular filtration rate, as well as changes in cytochrome P450 activity. In some cases, drug metabolizing and transporter activity will decrease, such as with CYP1A2. In other cases, for instance, with CYP2D6 and CYP3A, you see an increase in activity. And all of these changes need to be taken into account to adequately treat the mother and protect the unborn child from adverse events from the medication. Because it's difficult to study individuals during pregnancy, and you can't, there's ethical concerns about putting a child or an unborn child at risk by exposing them to medications that they may not need. There's feasibility issues of doing observational studies and opportunistic studies in some cases, and so it's really difficult to capture clinical data in pregnancy, and a lot of the data we have is very sparse and not of high quality information. So PBPK modeling is an excellent way of combining this data together, looking at the physiologic changes that a woman experiences throughout pregnancy, and being able to predict the effects of pregnancy on pharmacokinetics of drugs. And you can see that PBPK modeling obstetrics is not a new use for PBPK. It's been done since the early 1980s, but the number of articles has drastically increased in the last decade. In fact, there are now PBPK models in pregnancy for a number of drugs, and here's a list of the medications that have been published, or a partial list at least of medications that have been published. I'm sure there are many more than what have been reported in these papers and these reviews, but you can see it spans the range of drugs that are renally cleared, metabolized, different therapeutic indications. So we have a lot of knowledge that we've gained on these drugs, and how they're used during pregnancy and potential PBPK options. But one of the things I've been interested in is to see, some of these PBPK models have been developed based on non-pregnant data and not validated with pregnancy data. So as we're getting new data in pregnancy, how can we now go back, take the information from these PBPK models and see if we can reproduce them and confirm that the predictions they have made and the assumptions they've used are valid, and actually, learn and confirm the information that comes from those. There's been several publications written for best practices approaches for PBPK modeling in special populations, and this figure shows, the approach that's typically taken when modeling pharmacokinetics in a special population such as pregnancy. So the first step is really developing that adult pharmacokinetic model based on information from the literature, from your in vitro studies, from clinical data in healthy adults. So we develop the adult model, verify it, go back, adjust if we need to get a adequate, valid adult PBPK model. And then to determine the special population PBPK, we should simply just need to adjust the system parameters. This works for children. For pregnancy, there's some additional needs to add in, the fetal placental unit and distribution to that. But in general, the maternal parameters can be adjusted, and you should be able then to predict your PK in a special population. Now, to get information on these system parameters, you will then again go back to your literature data and potentially find some PK data that we can use to verify our model is accurately predicting. And then you can use that to simulate your study. And then the goal would be that your observations from that study would fit the model that you've simulated. So you can see here an example of where the model's been simulated and the data overlaid, and trying to see, are the study observations lying within a reasonable prediction interval. We're all aware of the learn and confirm paradigm that's usually used in PBPK model development, where you use PBPK modeling to understand what you have, capture the current best practice knowledge and data on a drug and a population, and then use that to make an inference on your new population, such as the pregnancy population, and then generate new data to test those assumptions. And so using this learn and confirm paradigm, we're able to take the PBPK models, integrate our data that we currently know and understand, and then understand whether, test your assumptions and see if it actually works in that background. Model reproducibility is a key step in validation, in that to grow our PBPK modeling efforts and to expand the inference and the knowledge from these models, we need to be able to reproduce the models and validate those assumptions, and then create new theories and hypotheses that can be tested and established in these models. But reproducibility is a challenge. I'm sure we have all been that person in the lab running an experiment and not being able to reproduce it or replicate it. So it's not uncommon, even within my own lab, to have something happen once and then we're not able to reproduce it. Even in the modeling world, if you're not carefully recording what you have done and the data you're putting into your PBPK models, you may not be able to recreate that model at a later time. One question though is there a reproducibility crisis? And this really expands beyond pharmacokinetics and quantitative modeling processes throughout all of science. This survey in Nature of over 1,500 researchers showed that the majority believe there is a reproducibility crisis. In fact, over 50% think there is a significant crisis. Many have failed to reproduce an experiment, either someone else's or their own experiment. And medicine is a very common place where reproduction is a difficult thing. In fact, some have even tried to publish a reproduction attempt, such as what we did here, where some are successful, a number of them are not successful. You'd think that reproduction in a modeling and simulation environment would be a little bit more straightforward and clean cut. However, as indicated in this study looking at quantitative systems pharmacology models from Kurek et al., they searched PubMed, identified 43 publications for QSP models. Of those, they manually curated the model code. They found that 9 had a pseudocode and were not able to be replicated. But 18 models provided the code. Of those, they were able to test 12 in the software they had available. Of those 12, only 4 were executable. And by executable, it means they ran without having major reason to search and troubleshoot the code. But they actually did not confirm that the results were similar to what the published results were. But even with this, a 30% success rate of models tested being able to be executed is a pretty sad state of affairs. So it's really important as we move forward and as we try to grow and build on the work of others that you provide sufficient information and details to allow your work to be reproduced. So I just have a couple of examples of case studies, the first being a study we worked on with betamethasone. Betamethasone is an antenatal corticosteroid given to individuals at risk for preterm birth to promote lung maturation in the fetus. It was originally studied in the 1960s in sheep, where they gave sheep a dose of 6 milligrams betamethasone acetate and 6 milligram betamethasone phosphate, and were able to see that fetal survival was greater than those that they had controlled. So they ran a clinical study in 282 women from 24 to 37 weeks gestation, where they gave the same dose and compared it to 6 milligrams cortisone acetate, which has 1/7 the potency. They dosed it twice, 24 hours apart, and observed the respiratory distress syndrome rate in neonates in the treatment group was 9%, which was significantly lower than that seen in the control group of 25%. So this study by Liggins and Howie in 1972 has become the basis for how we dose betamethasone and antenatal corticosteroids clinically in pregnant women today. So pregnant individuals are administered still a 12 milligram dose of betamethasone intramuscularly 24 hours apart for 2 doses. Recently, the question's been asked, are we overdosing antenatal corticosteroids? So we're using this dose that was determined in a sheep model. And we're seeing some response, but maybe we're overdosing the neonate or the fetus and having adverse events for them as well. So this was a simulation study that's been conducted using a betamethasone PBPK model, where they show that the typical betamethasone acetate and phosphate exposure in this red line has a very high Cmax, much greater than the estimated range for the therapeutic index of 1 to 4 nanograms per mil. And the same with dexamethasone, which is an alternative corticosteroid that's used in this population. And so you can see there's a much higher peak effect, which may then lead to increased risk to the neonate. So the goal of this studies by this group from Gates Foundation is to actually look at, can we reduce this concentration so that we can have a more durable response and exposure over the 48-hour period where we don't see these large peaks in betamethasone concentration. The PBPK model that they're using for this was developed by Alice Key and Mark Malad and published in CPT in 2019. And this publication, they describe the development of a full PBPK model for healthy volunteers, as shown here. They have both IV and intramuscular dosing of betamethasone in the healthy volunteer population. And then they verified the model in pregnant women using IV data only. They provide a nice table for their input parameters, which is recommended in many guidelines to help with reproducibility issues, so that you could just go in and take the parameter values you need from this table and enter them into software, in this case SimZip, and reproduce the model. And so we took this model, we added in their information that they provided here, and we tried to recreate it, mainly because we found that, well, they verified the model for IV dosing in pregnancy. As I mentioned on the previous slide, betamethasone is typically administered by intramuscular administration, and so it would be important to also look at data on intramuscular administration of betamethasone. And I will note that as of 2019, there was very little literature data and probably not good literature data on betamethasone concentrations following IM dosing in pregnancy, which is one of the reasons to incorporate this PBPK model. However, since then, there's been a manuscript by Franz Foysak where they reported the results of the B-Myelins trial, where maternal betamethasone was administered for prevention of respiratory distress syndrome in neonates, and they used a population pharmacodynamic approach to evaluate the data and look at giving some recommendations for dosing to improve personalized medicine there. This study was in a population of 178 women that were studied both for PK and then a subset for PK/PD. You can see the ages range from about 18 to 44 years, and most of them had some complication putting them at risk for preterm delivery, or had premature rupture of membranes or preeclampsia for which they were having delivery for. In this study, they administered the European formulation of betamethasone phosphate, betamethasone acetate, which contains 11.4 milligrams, 24 hours apart for 2 doses. And as you can see here, they collected maternal plasma, cord blood, and also amniotic fluid samples Here at IU School of Medicine, we have also conducted a observational PK study. This was led by Dr. David Haas, who's one of our OBGYNs here at the School of Medicine, who's had an interest in betamethasone for many years. Haley Trussell served as our research coordinator, and Larissa Lockie Silva was my postdoctoral fellow who helped do the modeling and simulation work here. This was an observational PK study in 242 singleton pregnancies. When the individuals were at least 18 years of age and between 23 and 34 weeks gestation. They were at imminent risk for preterm delivery. There were some exclusion criteria, including hepatic or renal failure and severe fetal anomaly. These individuals were administered 12 milligrams of betamethasone acetate and betamethasone sodium phosphate, the Celestone Soluspan formulation, which contains 9.95 milligrams of free betamethasone, intramuscularly, 2 doses, 24 hours apart. You can see here the demographics. Again, the age was similar to the FOISAC study, with women being 18 to 44 years of age. There was a mixed racial ethnic background here compared to the FOISAC study. So we took the model from Key et al., which was developed in version 17 of SimSip. At this point, we were using version 19 on a regular basis. So we initially started by putting the parameter values provided in the manuscript in version 19 of SimSip. And you can see here that there was a very minimal difference in the observations between version 17 and version 19. Our criteria for reproducing the model was that we would see less than 10% variability. This would allow for differences potentially in the patient population if we used a different starting seed than had previously been used, and potentially some variation in the version, but with minor consequences to the overall prediction values. So you can see for the data that was presented in the previous study by Key and Milad, we were able to pretty much recapitulate the concentration time profiles that they provided in the healthy volunteers for IV, oral, intramuscular dosing, and in pregnant women or pregnant patients for an IV dose. I will note that in their publication, they did change the CYP3A4 function in pregnancy to fit this equation. And so we did the same in version 19 of SimSip. So the important part of doing this was to say, well, now we have the IM data. Does their model predict what we saw clinically? And you can see from these plots and the ratio of predicted to observed, we did a fairly decent job. For our validation criteria, we were using twofold validation, which is pretty typical for a PBPK model. And you can see we fell within that criteria for both AUC and Cmax at the various gestational ages here. You will note that the data from the study at IU, which are presented in the first 3 columns, 3 sections here, 26, 30, and 34 week, was slightly over predicted, whereas the data from the FOISAC model was under predicted. And we looked at that a little bit more in detail, and there was definitely a difference between the 2 studies in the concentrations predicted, even after accounting for the differences in formulation. The populations were similar in age, gestational age, as well as weight. but ethnicity was different between the two studies, so that could play one role in the differences that we see here, as well as just the individual variability that's observed during pregnancy and of the PK of betamethasone. So the second example I have is in the setting of opioid use disorder in pregnancy. And here in the US, the rate of overdose deaths among women rose 20% between 2015 and 2016, and it continues to rise. Opioid use disorder diagnosis has gone up more than 4 times among pregnant women.
Legalize ricade.
And there's 4 times as many infants born with neonatal abstinence syndrome in 2014 than in 1999. So it's a major issue in pregnancy, and has some long-lasting effects on the neonate. Treatment of opioid use disorder in pregnancy either consists of methadone or buprenorphine maintenance therapy. Both are effective methods, but the buprenorphine dosing is a simpler method and easier for the patient. So methadone requires daily administration and visits at a licensed methadone clinic due to the high risk of overdose and mortality with methadone if it's abused. Whereas buprenorphine can be provided in an office setting here in the US by a licensed physician. And so it's much easier. The OB themselves can obtain a license to dispense buprenorphine. And so it's much easier for the patient to obtain and then can take it at home for the month before getting a new prescription. There's a similar risk of neonatal abstinence syndrome between the two, although it's reported that buprenorphine may have a slightly lower, shorter duration of abstinence syndrome. And while the neurodevelopmental outcome is reportedly favorable in methadone, there's less information on the long-term effects of buprenorphine. But in general, it's much safer for the neonate, the fetus, to be exposed to either methadone or buprenorphine than have the issues with the illicit drug use during pregnancy. So we had conducted a study of buprenorphine, an observational pharmacokinetic study in 34 Caucasian patients between 21 and 38 years of age in their 3rd trimester of pregnancy who were maintained on buprenorphine for opioid agonist therapy. As you can see, the individuals had varying dosing regimens for buprenorphine ranging from 2 milligrams to 20 milligrams a day, and had a wide variability in their pharmacokinetics. We did have data for both for buprenorphine as well as the metabolites that we collected in this study. So again, our goal was to utilize the PBPK models that had been reported in the literature to see if to validate them using our external data set. There was a previous model published by a group out of Pittsburgh that described buprenorphine pharmacokinetics following IV and sublingual administration in healthy volunteers. And then in later publications, they had extrapolated this to pregnant individuals. From my reading and knowledge of this study, it was well conducted, where they gave great tables with the physiochemical parameters, the distribution parameters, the clearance parameters, various enzyme scalars that were used to estimate the intrinsic clearance from microsomal data and so forth. However, once we started trying to actually reproduce the study in SimSip, again, we were using version 19. They had used version 15. We realized that the IV clearance that we were getting was quite a bit different from the clearance that had previously been predicted in version 15 of CMSIP. So we had a 30% lower clearance than what they had predicted in the publication. We thought, well, one reason for this is that we're using different versions of CMSIP. So sometimes the parameters will change between versions and the expression of level of an enzyme as we get additional information on a population or enzymes. So we dug out version 15 of CMSIP and reproduced it also in there. We actually got similar results to our version 19 study, so it wasn't due to variation in the software and changes in the population parameters, but there's some other issue that is preventing us from taking the information that was provided here in the literature and being able to reproduce it in CMSIP. We also felt there was some confusion in the description of volume of distribution. One of the compartments they noted was bone and additional with a value, and reading through this, they seemed that they added an additional compartment. However, as we further examined it, it seemed that they did not use an additional compartment, but this was just for the bone. KP was changed to 3.5. And then finally, we really had an issue with how they described the absorption model. CMSIP does not have a default sublingual dosing module, so they had created a custom dosing route using a inhalation and an oral form with a depot formulation or component as well. And they had given all these nice tables about how they did the sublingual dosing and where it worked, but we were not able to evaluate in CMSIP and understand how it actually went into the program and where the models were, where the parameters how they put the parameters into CMSIP. So we were unable to recreate that at all. I will note that the authors of the model were willing to provide additional details and information to us on how they did the modeling, but as the aim of this project was to reproduce the model based only on the published literature, that's what I am discussing today. So from that experience, we had several lessons that we learned. First, things aren't always what they seem. I had initially read the papers by the Pittsburgh group and thought there's a lot of data here, a lot of information that will go into a model. It should be easy to recreate. They're telling us exactly what they did. But as we started looking at it, it wasn't always consistent. Between the two papers, there were different parameter values given for some things. And the information didn't seem to be provided to actually integrate into the model. And I will note that three of us looked at this model with varying experience in PBPK, and we're not able to understand or to make it reproduced in our hands. So the details are really important when you're presenting your information and having the accurate details to put the puzzle together. So having also these details clearly defined and easy to understand. So I showed the two tables between the betamethasone study and the buprenorphine study. Betamethasone had everything in one table. It was really easy to pull out exactly the values we were given. Whereas for the buprenorphine study, we were going between multiple tables, and in some cases, the nomenclature used in the article text was different than the nomenclature used in the tables, and so there was just a lack of clarity that prevented us from putting the details together in this publication. So we came up with several recommendations from this project. First, it's, as others have noted in things, it's really important to specify the names and versions of software and packages used for model development. As I noted, there are differences between the various versions, such as model 15 may have given us different results than version 19, even using the same drug parameters. Authors should provide model codes or the files containing the saved workspaces as supplementary material, so that readers can go back and identify exactly where the parameters were, what they were, and where they were incorporated into the model. It's important to also include details of all the system parameters. As I mentioned, the populations often change between versions of the SimCip software, the parameters for those models. And so someone using a newer version may not have access to the default population parameters from a previous version. It's also important to list all your drug parameter values, the variability assigned, and the source of those parameters, or the calculation method that was used in a single table. One of the issues I've run into in other studies is that they may not clearly define what calculation method was used to, for instance, determine the volume of distribution of a compound. Finally, it's also important to include various details of the simulated trial design. These should be described as you would describe a clinical study, where you say, let us know the patient demographics, the dosing regimen, the length of the study, but also not just the number of patients simulated, but the number of trials simulated, so the total number of patients can be evaluated. And one thing that's rarely included in a lot of PBPK models is providing that starting seed, so that someone who's reproducing a PBPK model will be able to start with simulate exactly the same individuals that were used in the initial publication. Finally, it's important to discuss in detail the assumptions made during model development and any limitations of the model that you are using. So I'm not the only one to talk about this and note the importance of model reproducibility. There are several guidelines from the FDA, from the EMA, from OECD on using on best practices for documentation of PBPK models and the approaches that can be used to help facilitate reproduction. This chart comes from a manuscript from Ping Zhao in CPT in 2012. So this has been something that's been preached upon for quite a few years, and we're still really bad at it. So yeah, I'm just here to say, let's try to do a better job and see where we can get this reproduction to improve reproduction in our modeling. So one of the groups I'm that's working on this. So I'm as mentioned, I'm the principal investigator of the Maternal and Pediatric Precision and Therapeutics Hub Data Model Knowledge and Research Coordination Center. This is an NICHD funded network with Indiana University and Ohio State University leading the coordination center. The University of California, San Diego, and Vanderbilt University Medical Center have clinical centers of excellence in therapeutics, where they're actually doing some great research study looking at antibiotics and the maternal milk infant triad and real world evidence in pharmacokinetic related to opioids. So one of the aspects of our center is a pharmacometrics and clinical trial design core led by Rob Bees from the University at Buffalo and myself. And the goal of this core is to bring together expertise in pharmacometrics and clinical pharmacology and clinical trial design in obstetric and pediatric populations from a variety of individuals. As you can see here, I have pictured the core members of the IU Buffalo Pharmacometrics Clinical Trial Design Corps, as well as a variety of our external advisory board members and consultants. But we also are working with the other centers within Imprint, UCSD, and Vanderbilt University, as well as reaching out to other groups, currently Cincinnati Children's Hospital and the University of Washington. But we're looking to actually expand this to other groups and networks. And one of the goals of our center, in addition to bringing together this expertise, is then to help develop best practices and guidelines for conducting pharmacometric studies and PBPK, PD modeling in maternal and pediatric populations. With that, I would like to acknowledge those who've made this work possible, including our my fellows, Larissa Lockie-Silva and Rebecca Silvola, who were instrumental in the manuscript that I was discussing today on reproducibility. Andrea Masters runs our clinical pharmacology analytical core lab. Misty McDowell and David Haas were clinical collaborators running the opioid and Betamethasone study, as well as our research coordinators, variety of funding sources for and the patients and their families, without whom this work would not be possible. With that, I will thank you and take any questions.
Thanks, Sarah. That was really great. Amin's the first one to jump with his hands up. So did you want to speak, Amin?
Yes, thank you very much, Sarah. We loved, when the publication came out, and we love it even more now that you have explained all the details of, what has gone, to that particular publication and piece of work. I have got actually 3 questions. Number 1 was related to if in any of those papers that you were looking at, the reports that people were publishing, you had the workspace already available, that you can actually get the workspace and just plug it into what you are doing. What percentage of them they gave the workspace, or at least one workspace of the variety of the things that they had done?
Neither of those publications did give the workspace.
Oh, that's bad then. Well,
I have to say, I don't remember if we looked on the SIMSIP website to see if they were available, but I don't believe they were at that time.
Okay, so I think, we have to encourage people, to at least put, I know that sometimes, they are doing more than 1 or 2 simulations, we are not expecting every workspace, at least the central workspace should be there, that's one. Okay, the next one that I was going to ask is that how easy or difficult for you was to actually based on the initial question that you asked with regard to the 3A4 changes, to monitor the version X to version Y changes, because when a version comes out, obviously there are some notes that the R&D team put together and basically say, okay, this is the executive one, but this is the detailed one. It basically says that what has changed from sort of version X to Y. So how difficult was it for you to actually to find them? Because if it is, then we have to do something about it.
Yeah. So for this paper, I still had versions 15 on my computer and was able to get an activated license for it. So I was able to go back directly to the software and look at it. Actually, we had an experience yesterday where one of the models we're looking at reproducing now is in version 14, and I don't have access to version 14. So I'm hoping that Oliver will be able to come through.
But that is one thing is having access and one by one going yourself and actually teasing out what was different between 14 and 15. But the other one is actually the list of the changes that that
Exactly.
comes when the version comes out, because that is published. That is basically says from version 14, now version 15 is out, we would like to tell you that these are changed to this and for this reason, that reason. So you are saying that's difficult to find.
So I know it's there. It's easier for me to go back to the version
and then check it yourself.
and
Okay.
check it. The other problem is if I'm going from version 15 to version 21, now I may have to look at
Correct.
multiple of those files.
Correct.
And
Every year by year.
Every year by year.
Okay. And the last question, this might be brief. Have you got, or your group has got, any experience with regard to the reusability of the models? So this is not reproducibility, but reusability. Have you actually checked, followed, to see how the reusability, sort of scores? How many of the models that originally produced are reused later on?
I have not, like in publications, to see if there's other publications on the models. That's not something I've looked at.
Okay, good. All right. So that's good for us, because we are putting something out on it. Two of my students in CAPCA have done it, but we are just submitting it in a couple of weeks. Okay. So it was very interesting also to look at that. But hopefully I will share some of the slides that they have got. But that's coming out. Okay. Thank you very much. Enjoyed it.
Yeah, and I'll go back to the putting out the workspace and your comment about not needing to put out every workspace. And I agree with that, because even putting out this manuscript, we put out the workspaces with some of the workspaces we used for it, but it was very limited. We obviously had gone through a lot more trials for the buprenorphine model, and I think we put out 1 or 2 workspaces for that, just to show this is what we tried, and this was our best fit or our best option. So I do know it, even within my work, the workspaces and tracking them can get very, very difficult. So but I do think it's important to put out at least one so that you have those data. And that's another place where you can find the when that workspace is put out, you should put out the population data too, not just the drug level data, which would then allow you to go back and pull the population values.
Thank you.
Thanks. Ian, did you want to ask a question?
Yeah. Hi, my name's Ian Templeton. We actually had adjacent posters at a meeting a long time ago when we were both postdocs. So it's really exciting to see you again. And thanks for the great talk. My question actually is more of just kind of a comment. I completely agree with what you're saying, that it is hard to reproduce the models. And I think, at least in my experience, part of that has been the limitations of the manuscripts not allowing us to include all the information that we want to include, and the reviewers being more focused on the kind of high level stuff than the modeling details. But one of the things I've noticed, at least in the CPT PSP, is they're letting people put everything in as supplemental. So I think like what you say is true, but I also think part of the issue is the journals and just kind of getting the word out that people need to like know that this is an option, that they can submit like all the details separately. Yeah.
I don't think that made it into my slide. At one point I had the information from the CPT PSP journal about encouraging you to do that. So yes, I think it's important that the publishers also push that we make these available in supplements. There's also the ability to put it in a GitHub or another repository that would be publicly available as well, if the journal doesn't allow for the supplement.
Yeah, that's a really good idea. I guess a lot of people on this meeting too are reviewing these articles as they're submitted. So I think all of us just have a responsibility to kind of suggest that if we're not seeing it included, suggest that people include this in the supplemental information.
Yeah, I've actually, one of the suggestions at meetings on reproducibility is that the reviewers reproduce the models, but that's also difficult when we may not have access to all the software that's used or the expertise to reproduce it. So, as I say, the buprenorphine paper I thought was very well written, and I was actually surprised when it didn't reproduce because it looked like the information was there.
Yeah, great. Thank you. Thanks for highlighting this topic.
Thanks, Ian.
Hi, thanks. Sibylla, you're next.
Okay, first of all, thank you very much. I enjoyed it. I'm one of the naughty person who actually likes reproducibility because it saves me time. And I have several comments and also some questions. The first comment would be that whatever has been changed in the different versions, in the appendix of the help file, they are listed for the population as well as for the compounds. So therefore, even in between different versions, and we keep that up. So there's always one table per version, and we keep that in. So in the appendix, you find these changes. If an enzyme has changed or anything, it can be found there. The second one is you had now an example, version 14. That's now your next problem, right? Version 14 and run it in version 21. You have something submitted to the regulators, and then they have the great idea. How does that work in version 22? Right, we are aware of that. So one of the things is, you mentioned that you do need to go version by version to look for changes. You don't. Because the first thing that I would suggest is you take the workspace in version 14 and open it in the latest version. You will have there one slight issue. You will need to go via version 17 to open it up, because there we had like a clean-up version. So version 17, you can open in version 22 and things like this, but version 14, you need to go via version 17. You should be able to have version 17 at the moment on your computer. If that is not the case, send it to us. We make a version 17 file, and you can open that then in version 22 or 21. So you don't need to go version by version. The other comment was, and now I'm getting actually to a little bit more the members area. We have on the members area by now more than 80 compound files that have been submitted. These are not our compound files, these are submitted from publications. In order to come on the members area, we want to have it in the version it has been published. So really the file they have done the publication with. And also when I request now all paper, the version, please submit it then in version 12. Submit it in version, I don't know, 9 if you have had it. Because we can actually check that. We open it in that one, and what I do is check that whatever is run, it's actually also in the publication. That means the input values are there, as well as the profile that comes out is there. When I did that, from the 80, 3 of the compound or 3 of the submissions have never been released, because the input value for the enzymes was not as it was published, but I got the profile as it was published. So it's obviously no one will be able to reproduce that file ever. What I do, I go back to the author and point that out. So and then I leave it to the author if they do an erratum or what they do as a consequence out of it. The other thing is, and now I'm coming exactly to that what you said, pregnancy, other populations. It's a different thing. And how many workspaces should be uploaded? If you just do a compound file, healthy volunteer, don't change any population data, the simple compound file is enough. If you change something in a population, I would prefer a workspace, because in that moment you have changes done on the compound and the population. Honestly, if you use the normal compound file and you only do the population, submit the population. With pregnancy ones, it's a special thing, the same with the pediatrics. I do not agree just one workspace. I would need to have then the workspace for each compound with a fixed population. And we go through that exercise at the moment with Khalid's latest paper. He gave me 44 workspaces to check through in order to put them then on the members area. So we will figure out how to put actually a combined part on that we also can trace these ones. So the aim of our repository is that it matches what has been published. And I think, and also that we are internally checking it once. When you're looking actually at the members area, you can see sometimes there is an extra comment, like a solubility value of this and this has been added. This extra comment is only in because that is not part of the paper. And without that, you can't reproduce it. So, and that is maybe a hint for you. If you can't reproduce a paper, look at the formulation, because that's normally the one where the people are not giving the information on. The general thing, KP scaler and everything is there. It's these small things which is actually then missing on there. And for that, I mean, everyone who is there and have a paper, I would appreciate if you submit the files. And both of the ones you had looked, I just checked it, are not on our members area. They have not been shared. And I have not contacted everyone, but I have contacted a lot. And normally what I get as response, if they are not shared, is because the project doesn't allow it, which is for me a little bit worrying, because the publication is allowed, but sharing it on a members area where people actually can reproduce it is not allowed. So that's something I would just say, if more people would submit it, that would be really, really useful. So what do you think? And now I'm coming to a question to you after all of this. So what do you think would be the best kind of repository? A general one from basically SimSip would not be involved, a general somewhere where you can compare different models also, right? Different platforms, or that we are actually extending our database, and what can we do on our members area to make it more appealing? Because when you tell me you haven't even looked there, you rather spend your time in trying to reproduce, then apparently we are not doing a good job with communicating it. Or is it complicated to get things from there? What is the reason?
I think I probably actually did look for these when they were out. I just haven't looked recently, is what I was saying. And I don't know if the fellow who's working on the current project has looked there, but they are also coming from, what we're working on right now is coming from FDA reviews. So it's not published necessarily. So I doubt those are probably the ones of we are not allowed to release it to the SimSip platform. From a I appreciate what SimSip's doing, and I think the idea that you're actually validating the model before you put it out onto the platform is really encouraging too. And I think that's great for those who are SimSip members. I think, from an open science standpoint, having something that could be accessible by others would be important as well. So, if someone's reading it and doesn't have access to SimSip, they would be able to get additional information on the parameters and so forth. I think would be helpful too. So maybe a little of both.
Yeah, I think, the paper obviously should always have all information included, but without the simulator, you would not be able to use it anyway, so the workspace. So therefore I'm struggling there a little bit to see the point in it, because you need to understand the mechanism behind it and also what you put in. And the value that you put in SimSip is not necessarily a value you can put in G+ or in PKSim, because the algorithm and the units are different. So therefore I don't know what really a general platform with all of these would help, and who would actually make sure that what's on there is right.
Right, and that's one of the things we're struggling with from the imprint standpoint is, we can identify models, we can put them in a database, but it's the confirming that they're accurate is a little bit more difficult. So.
Well, thank you very much.
Thanks.
There's what Armin's been very patient waiting to ask a question, so we'll let him ask a final question, if that's okay.
Well, many thanks, and really impressive, and I can commiserate given the effort you had to put into trying to reproduce data which should have been reproducible in the first place. Just by way of comment. Well, 30% success rate seems to be a huge improvement. It was reported to be about 5% 10 years ago. That's when the biomodels database was set up, and that could serve some kind of example, perhaps. They look at the QSP models and run them through, and kind of put a seal of approval if the plots reproduce. They don't make an assessment whether it does or doesn't make sense. But obviously, that all runs in SPML, and simulator. Well, in a way, it's better that it's more standardized framework, but yeah, accessibility can be a bit of a problem there. So that's all I had to add at this point. But really very encouraging that you really took the time and effort.
Thanks.
Thanks. Well, thanks again, Sarah, for presenting today. I think it was a really important topic, and yeah, it got a lot of comments and questions, so that's really good. Do you mind just going to the final slide? I'll just highlight. We're actually staying on the topic of pregnancy, and we're going to be looking at pregnancy and lactation PBPK model, recent advances and applications. So Amita is going to be presenting on that in October. So please register online on the members area if you want to join that one. Okay, so thanks again, and take care. Have a good rest of your day.
Thank you.
Thank you.
Thank you.